Hi everyone, welcome back to iClick's Biology Discuss. In the previous class, we have discussed about the detailed structure of prokaryotic cell. Okay, so in this class we will discuss about the mesosome and the ribosome. Okay, so first let's go to mesosome. Okay, see here, this is the infoldings of the plasma membrane. Okay, the outer covering, that is the capsule. Inner to capsule there is presence of cell wall, and inner to cell wall there is presence of plasma membrane. Suppose this is the plasma membrane, and this plasma membrane sometimes shows ingrows. Sometimes it shows ingrows or infolding inside the cytoplasm. This ingrows or infoldings of this plasma membrane, it makes some vesicular or tubular structure, and this vesicular tubular structure, that is called as what? That is called as mesosome. Okay, and it is believed that, let me first write this. Mesosome, this is the vesicular and tubular structures made by infoldings of plasma membrane. Okay, so this is the mesosome. So in case of some prokaryotic cell like your bacteria, there is presence of this type of infoldings. But in case of eukaryotic cell, the mesosome is absent. Okay, and it is believed that the mesosome, it is helping in the respiration process. Why this is believed that? Because some enzymes which are responsible for the respiration process, that are present on the mesosomes. That's why it is believed that mesosome may help in the respiration process. Okay, it is believed to be helps in the respiration process because the mesosome contains some enzymes or the respiratory enzymes. These enzymes are the respiratory enzymes. Okay, and see here, the mesosome is attached with this DNA, that is the genetic material of this prokaryotic cell, so that it is also believed that the mesosome may contain some enzymes which may help in the DNA replication in case of prokaryotic cell. Okay, so another function of this mesosome, that is it may help in the DNA replication process. Okay, it may help in the DNA replication process. So this is all about your mesosome. Okay, so mesosome helps in the respiration process. But in case of your eukaryotic cell, as you know that in case of eukaryotic cell there is presence of mitochondria, and this mitochondria helps in the respiration process. But as we know that in case of prokaryotic cell there is absence of this mitochondria or the membrane bound cell organelles. That's why the mesosome, that is helping in the process of respiration. Okay, next go to this ribosome part. Okay, ribosome, this is the only cell organelle present in case of your prokaryotic cell. But this ribosome, that is not a membrane bound cell organelle. This is not a membrane bound cell organelle. Okay, this is not a membrane bound cell organelle. In case of your prokaryotic cell or eukaryotic cell, there is presence of ribosomes. In case of prokaryotic cell, the ribosome is the 70S type of ribosome. This is the 70S type ribosome. Okay, if we see the composition of this ribosome, then mainly this ribosome is made up of two things only. One is the RNA, another is the protein. So this protein may also call as ribosomal protein. And this RNA, that is called as ribosomal RNA, or in short form you may write this as rRNA. Okay, but if you see the percentage, then the RNA, that is having 65% of RNA, and protein, that is having 35%. Okay, 35% of protein and 65% of RNA, that is present in case of ribosome. Okay, as you know that the 70S type ribosome, if you see a ribosome, that is mainly made up of two subunits. One is the smaller subunit, another is the larger subunit. Suppose this is the ribosome and it has two subunits. One is the smaller subunit, and this is the larger subunit. Okay, and the smaller subunit and larger subunit, they both are made up of what? They made up of proteins and the RNA. Okay, if you see the composition, that is made up of proteins, these are the proteins, and there is also presence of RNA. Okay, and suppose these are the proteins, and in case of larger subunits, two types of RNA or two numbers of RNA is present. Okay, and this RNA, that is 23S RNA, whereas this RNA, that is 5S RNA. The sedimentation coefficient of this RNA which is present in the larger subunit, one is the 23S and another is the 5S. Okay, so this is the RNA, and in case of your smaller subunit, the ribosomal RNA which is present, the sedimentation coefficient is 16S. Okay, and in case of your smaller subunit, there is presence of 21 number of proteins, and 34 proteins is present in case of larger subunits. Okay, so this is the ribosome which is present in case of your prokaryotic cell. In case of ribosome, there is 70S type ribosome. But if you see the smaller subunit and larger subunit in individually, then the smaller subunit, this is having 30S, the sedimentation coefficient is 30S, whereas the larger subunit have the sedimentation coefficient, that is 50S. Individually, they have different sedimentation coefficient, but when they undergo the process of protein synthesis, both of them, they join with the RNA for the protein synthesis. At the situation, the sedimentation coefficient is 70S. Okay, when both of the subunits, smaller and the larger subunit, join, at that situation, the sedimentation coefficient is 70. Whereas the individually, if you consider them, then this is 30S and this is 50S. This is the larger subunit and this is the smaller subunit. Okay, so if you see, this is the normal structure, this is overall structure of a ribosome. If you see a detailed structure of a ribosome, how it looks. This is the 30th subunit. Okay, this is the smaller subunit, and these are the some parts of ribosome. This part is called as platform, and this is called as flap, and this part, that is the head part, and this part, that is the base one. Okay, and there is larger subunit. There are three different parts. Okay, this part is the ridge, this is central protuberance, and this part is called as stalk. Okay, when they are individually present, this is the 50th subunit. When they are individually present, they look like this. Okay, but if they are joined during the time of protein synthesis, it will look like this. This is the 30th subunit and this is the 50th subunit, and they will make 70th type of ribosome. Okay, hope you understand this clearly. In this class, we have discussed about the mesosome and the ribosome. Mesosome is the site of respiration, whereas the ribosome, that is the site of protein synthesis. As you know that the protein synthesis in case of prokaryotic cell, there is requirement of two things mainly. One is the ribosome, another is the RNA. That is mainly mRNA, and the ribosome having the rRNA. Okay, and what are the composition of this ribosome? That is the RNA and the protein, and maximum percentage of this ribosome, that is the RNA, whereas the protein, that is 35%. Okay, and in case of prokaryotic cell, there is presence of 70S type ribosome, and if you see the ribosome, then there is presence of two subunits. One is the larger subunit, another the smaller subunit, and the smaller subunit, that is having the sedimentation coefficient, that is the 30S, whereas the larger subunit, the sedimentation coefficient is 50S. So this is the second part of the detailed structure of the prokaryotic cell. In the upcoming classes, we will discuss about the different parts like the pili and the flagella and other parts like your nucleoid or the xenophore, that is the nucleus part of in case of your prokaryotic cell. Hope you understand this clearly. In the upcoming class we will discuss other things. Thank you. Please like my channel, share and subscribe. Thank you.